So I've been asked to talk about whole genome sequencing. So if we just go to the next slide, in terms of what I'm going to cover, well, we've kind of covered all this already. So we were talking about vision before. This will be the revision because I'm going to go over it all a little bit again. So what is the genome, and then what is whole genome sequencing? And there's a bit of a clue in the name about what we're going to be talking about. And a little bit about how can it help us in sort of cancer and specifically in sort of salivary gland and rarer tumours as well. So I'm glad Sam/Rob didn't use this analogy that I use, because I like the analogy of thinking of the genome like a recipe book. So you can imagine that every human has their own recipe book for how the body should function. And it's this be-all and end-all of recipe books. It's got every single recipe you can imagine for whatever occasion you need, it's there. Half of the recipes come from your mum, half the recipes come from your dad biologically. So you get this combined recipe book, which is unique to you, the combination of two different families. And similarly to how you wouldn't take, I don't know, a vindaloo to a christening, you take a celebratory cake. Each occasion will need a different recipe from that recipe book. And similarly in our body, the heart will be using different recipes to the lungs, to the salivary gland, etc. So while every cell in our body that we were talking about has this copy of the full recipe book, the full genome, they pick and choose which ones they need in order to do what they need to do and do the function of the body. And all together it comes to be able to do what we need to do in the body. And as we've been talking about, the way most of it functions is by going through proteins. So this is a recipe book which can be for anything, but in the body it would make a protein which then goes and does something. So as we've kind of touched on already, the body has its own genome, but if you're developing a cancer, that could have its own genome. So one of the ways that cancer can form is by having an error in that recipe book. So if the recipe says add a teaspoon of sugar, but then there's an error when it's being copied and it says add 10 teaspoons of sugar, it's going to be a different outcome. A different recipe will make a different cake. And similarly, if the cell is different because of an error like that, it might function very differently. It might grow quicker. It might do things that the cell shouldn't be doing, and that could lead it to become cancerous. So when this kind of copying is done between the cells, if there's errors in the new cell, that can become cancerous and it changes the behavior. And when we're thinking about the genome, we think about the human genome, and that's termed the reference genome. So that's just this stylized picture of an average human here. So when the Human Genome Project was being done in the early noughties, they wanted to find out, well, what is the human genome? And we talk about this reference, so it's kind of the average human has this genome. This is the blueprint for a human, and we call it the reference. And there's a lot of debate about how good that reference is. I think that the reference genome is mostly made from about 8 different humans. So you're not really getting all the variety of the different humans and ethnicities that we have thousands of different copies of. But you can imagine we have this reference genome, then we have you, us, the individual person, and then as we were talking about, if we develop a cancer, that cancer could have a different genome. There's the reference, which we kind of assume is the normal. We have the person, that's our individual one. Then we have our cancer, which has its own genome. And we're kind of talking about this. If you were to print off the whole human genome, it's a massive undertaking, and I don't really know why lots of people love doing this, but there's loads of these copies in universities around the world, and when you open up one of these books, it's just this tiny, tiny font of all these different letters, and it's very, very big. There's 3 billion letters in the human genome, and that codes for how we do everything in the body. And traditionally, when we're testing for genetic changes, as we've been talking about, we're really just looking for, well, what we think we need to look for. So we look for one change that we think will be useful for us. So if we're using the recipe book analogy, again, we'll flick to the right recipe and we'll look at line 4, because we know that line 4 often leads to problems which we can get information from. So we'll do it very specifically. And increasingly nowadays, we don't just look at one area, we look at a group of them together. So at the Christie, for example, in our typical panel, when we do genetic testing, we look for about 30, 40 genes, I think, in salivary glands. So we'll do them all at once, but we're still only looking at a small number of things in total. And are targeted, they're the ones we think we'll get the most information out of, but it's still fairly small. And sometimes, just to finish, we'll test big regions of the genome. So we'll do a chapter, we'll do a couple of recipes instead of just these specific lines. So whole genome sequencing then is taking the other approach, and instead of looking for things we want to find, picking what we think will give us the most information and doing specific tests, it's, well, let's just look at everything. Let's do the whole genome. And as you can imagine, we then pick up a lot more information. So this is the entire recipe book being analysed. And if we're doing this, as we mentioned before, we'll be testing the cancer specifically in this setting, the whole genome of the cancer. But we'll also want to be testing the germline sample. So that's the person, the patient sample as well. And we're comparing differences between the patient and the specific cancer, but also we can compare back to the reference genome as well to find these changes that the person has that they may have inherited from their family. And so, what are the advantages of this approach where we're doing the whole genome instead of just targeting different things which we think will be useful? Well, as you can imagine, we're not just thinking, well, I think we'll get information from here, so we're going to look there. We're not just looking at the areas that people commonly look at because people commonly find things at. We're looking at everything. So we get a lot more information here and we can find some rarer changes. That's important because we may not know about some things which could happen, and if we're doing this whole genome, we can find these things. But also in certain cancers, we don't know as much about them as we do in other cancers, and especially in rarer cancers. We don't know as much as we would in such as breast cancer, which we have a lot more cases of, we've done a lot more research on. If we're doing the whole thing for these rarer cancers, we make sure we're not missing out on things, we're picking out on things we want to pick out on. So you get this extra detail, and importantly, the traditional testing that we do is looking specifically at sort of errors in the letters. So if you've got letters which are coding for a certain thing, the traditional testing is just seeing, well, are there any error in those letters, just a specific letter change? But there's a lot more information we can get from the genome. For example, those fusions we were talking about, fusions would be picked up very easily in whole genome sequencing, whereas in traditional testing, we have to do specific fusion testing for it. And as we've touched on, you're testing your genetics as well as your cancer's genetics. So you get this inherited information, which is positive because in some things they're very useful. There may be a mutation in the family which predisposes you to cancers or predisposes you to impacts from different treatments, but it does have its negatives as well, which we'll come to. And as we've touched on, research is a big thing here. So if we're doing this whole genome sequencing, we're finding things we didn't know we'd find. That opens up the door for research and things which we can look into a bit more. And that, by the end of the day, will hopefully help us discover the next steps we need to do to improve the care of these types of cancers. And we've kind of already touched on this already, but if you're thinking of doing genetic testing, you can look for things you can try and treat, or you can look for things which can try and predict how the cancer will behave. It may be that a specific change indicates it might be a little bit slower than normal, it might be a little bit faster growing than normal. You can try and get this information from these testing. And yet, if we can get more information, we can plan our next stages of research. And I guess at the end of the day, a lot of the reason we do genetic testing is to give us information for research, to give us an idea of the diagnosis of the cancer, the behaviour of the cancer, but really what we want to be doing is getting a new generation of drugs to treat the cancer. And as we were talking about, the ideal approach would be to personalise our medicines specifically to you as a patient, you as a person, and your cancer specifically. So there's a lot of work going on at the minute to do this genetic testing, but also to find the treatments which can go alongside it. And in the last few decades, we've generated a lot of treatments which specifically target genetic changes, which we think are pushing the cancer forward. And if we try and block that, it stops that sort of accelerator and it tries to pull back on the cancer with some really good approaches. And a lot of these medications exist, and if we find them in rarer cancers, we can think, well, would it be beneficial to give one of those drugs from other types of cancers to this rarer cancer? And we can't always be sure that it will work the way we expect because we don't have that evidence base behind it, but it's the first steps in trying to provide that evidence for it. And also, as we were sort of touching on clinical trials, increasingly to get on the trial, they want you to have a specific change because they're trying to target their drug to a group of patients with specific changes. So if we're doing whole genome sequencing on a cancer, then we may find a change, and there may be a clinical trial which is looking for things with that change, and then we can try and match to clinical trials. And at the Christie, on our experimental cancer medicine unit, that's been going on for a while about this specific genetic testing, not for certain things that we think we might find, but can we find anything which might target a clinical trial in the future. So we've been doing that at the Christie for some time, not using whole genome sequencing, but using just very big panels of checking for lots of different genes. A lot of clinical trials will say, well, we need you to have this, so if you come to us, we'll test you for that one thing. But that's a bit of an inefficient approach, and there's a lot of work going on to say, well, instead of just saying the clinical trial checks for A, the next clinical trial checks for B, the third clinical trial checks for C, can we just test the cancer, do whole genome sequencing, and then we'll find A, B, or C, and then we can say, you know what, go straight to that. We've done this one test, which will give us all the clinical trial information. I think a group in France has been looking into that, and they've had some really good success about doing whole genome sequencing on patients looking for clinical trials, finding these things which they can then take to the drug company and say, I found one of those patients you were looking for, which has this change when I did this genetic test. But as with anything that has positives and advantages, we have to think about the disadvantages involved as well. So one of the benefits of doing whole genome sequencing is you can get information about the inherited side of things, your own genome, and that can be very relevant about your risk of cancers, how you'll behave on certain treatments. But you could also pick up a lot of unexpected things or things which aren't relevant, which could cause problems. An important thing in genetics which we have to think about is, while I could do a test, the information that comes from my testing could be relevant to the rest of my family who didn't consent to have that test done. And if I'm found to have a susceptibility to breast cancer in the future, then my sister and the wider family would have a lot to know about that information, more so than I might have to know about it. And it's something that we can't, when we do this test, we will find that information potentially, and there's ethical implications of that, and something we just have to be mindful of. There's also some talk in America about insurance companies trying to use our genetic information to change the way they charge patients for their insurance premiums, which I think isn't allowed in the EU and Europe, but it's something which there's talk about trying to better target genetic information. And so doing this tests are great, but they may have issues. Another thing about whole genome sequencing is, because it's a very specific test which needs specific information from it, it's very precise. Typically when we do genetic testing, we use a really old sample from surgery or something that's existing in a piece of wax somewhere. But if we want to be doing this very highly specific test, we need a new biopsy ideally. We need to freeze it straight away so we can use the sort of fresh sample frozen instead of it being interfered with by the wax. And that involves doing a new biopsy, which has its own risks. There's logistics of it because it has to be frozen and sent to a laboratory who can analyse it pretty quickly. There's a lot of logistics in doing it, getting it delivered, the delivery company, the FedEx with the freezer, etc., and making sure that you're not losing it en route, because then it's a wasted sample and we can't actually analyse it. The laboratory has to be very specific, so Manchester Royal Infirmary has a big genetics suite, that's the hub for the region. They can do it, but not every laboratory can do these big testing. And then it's got to be interpreted. You get a load of information. And when we first started doing this, we just didn't know what to do with the information. And now we can only do it with things like machine learning and artificial intelligence that are starting to analyze these things. We're now being able to do it easier, but it still involves a lot of analysis, a lot of work to sort of see the wood for the trees, like when you're doing this massive information. And then, is it relevant? So you can do the testing. If you do whole genome sequencing, you'll find a lot. If we go back to that reference genome, the reference genome has about 3 billion letters in it. And a typical human will have about 5 million changes compared to the reference. And that's why we're all different. That's why we have different hair colour, et cetera, et cetera. But because of that, if we do test someone, we will find changes. But it might not be relevant in the slightest. It might just explain why you're slightly taller or why you have brown hair, not blonde hair. And it might not actually help us clinically. So we can do this test, but we can't guarantee we'll get anything changed at the end of it. And so what are we doing in the NHS? Well, there's been a big push in the last decade to try and do more genetics in NHS and sort of prepare the country for the next stages of medicine, which we think will involve a lot of genetic testing in cancer and outside of cancer. So there's been a big push from government and a big push in the NHS to fund these testing samples and build up the capacity. If there are areas of the country which can't do this, try and fund it so they can do it, so we can give this equitable access to these schemes across the country. So Genomics England has been set up, this body designed to try and promote genetic testing in the UK. And there are these regional hubs all across the country to sort of standardise the procedures and make sure patients can access them. Our one for the Northwest is here in Manchester, a mile or so away at Manchester Royal, but every area has a certain one. And there's now a big book or a big Excel document online in the NHS which says you can do these genetic tests for this. And if you meet the criteria, you're welcome on the NHS to then have this genetic testing. And we're starting to try and introduce whole genome sequencing into this. So specific cancers are now eligible for whole genome sequencing, as are any other cancers which have limited treatment options, etc. There is this option to go forward in the NHS. But of course, we need to have the capacity to then do it, and that can be a bit of a stumbling block at the minute. But it's something which hopefully, as we get more experience in doing it, we can do a lot more easy in the future. So how does it work? Well, because we were talking about how we're getting a lot of information about your genome and your cancer, there's a consent process we need to think about. It's a bit different to the typical genetic testing because of that relevance to your family members, etc. So there's a consent process that has to be done where we have to explain about, well, what the implications might be if we find anything, and we might not, but if we do find anything. So consent has to go in first. Then we need the biopsy. So typically in a cancer patient, it'll be a biopsy of an active area of cancer in the body, for example, in the liver or wherever. And also because we're testing your own genome as well, we typically do a saliva, sort of a rub on the mouth, on the lips to get the germline sample as well. Then it goes off to the lab and they do the actual analysis on the fancy machines and then take the information to the specialist sort of software to analyze it. When they first did a human genome, I think the analysis suites, like the size of this room, these massive machines, there's like 100 groups all around the world, which took 5 years or so to process a human genome. And now the machines are like the size of this desk, and you can do it in like 10 hours, a bit under a day, and it's much cheaper now. So this technology is going forward at a rapid pace, and it's allowing us to try and use it more and more to try and improve research. And then, yeah, a bit about the Christie. So I know that Rob and the Christie have been interested in this biobank of samples for research, especially in salivary gland cancers, which are fairly rare. And if we can get more information about these cancers, then we can take the next steps of research forward. And traditionally, the biobank was using information from previous samples, from surgical samples, getting access to that and doing research on them. But at the Christie, we're also starting to try whole genome sequencing on patients with salivary gland cancers. So, as we've mentioned, we need to do new biopsies, we need to do the consent process, etc. But when we've done this testing, I think we've done about 15 or so now, the whole genome sequencing. As we've mentioned, in the majority of cases, we haven't been able to find a specific thing to change our management. A lot of what we've been finding has been what we've spoken about before, these gene fusions, which are very common in certain types of salivary cancers, sort of what we'd expect to find, as you can imagine. But we have had a patient who we did find a mutation, which we thought, well, this might be relevant for a drug treatment, or it might open up a clinical trial. We've been able to start them on a targeted treatment, targeted to what we've found on the whole genome sequencing. And that patient had already had traditional testing. We didn't find anything there. When we did whole genome sequencing and looked in more detail, we were able to find something and offer them a treatment. And we also found a few where we can potentially offer them clinical trials in the future if a clinical trial were to come up looking for that certain thing. But overall, we're increasing the understanding of genetics, which will help us understand salivary gland cancers, which will hopefully help us work on the next generation of research and treatments for salivary gland cancers. So sort of to summarise, how can whole genome sequencing help us? Well, it is early days, and there's been a lot of work to try and integrate this into clinical practice in the NHS, and there's still a lot of roadblocks along the way. It's a lot more complicated than traditional genetic testing. You need the new biopsy, you need the consent process, you need the lab to be able to do it, you need to get it there quickly, you need to do all these extra things. But things are changing very quickly. The technology is getting better and better. The infrastructure is building up more and more. So it's starting to become more easy. And that's giving us more information for research about how the cancer behaves, why it's doing what it's doing, trying to find targets for future drugs. And at the end of the day, our overall aim is to try and match patients to either clinical trials to get evidence for future treatments, or try and use existing treatments, such as we were talking about with the NTRK fusions, that we can apply to new settings. And that's relevant in rare cancers because the clinical trials, which typically want thousands of patients in them, are harder to do in patients where with the finding thousands of patients with the same cancer is more tricky. So I think that's the end, yeah. So thanks.
Thanks for
It's a
that.
bit of a whistle-stop tour.